Welcome to the Explainer. Today, we're not just looking at your standard laboratory tool, we're actually looking at a portal. The compound microscope is quite literally our spaceship to another dimension. It allows us to step totally out of the macro world we live in and explore this hidden, invisible universe that exists all around us, and, well, even inside of us. So how exactly do we explore worlds that are just way too small for the human eye to even perceive? It's a fascinating question, right? And the answer is actually an incredibly elegant combination of three things: light, lenses, and a precisely engineered structure all working together in absolute harmony. Okay, let's dive into this, because before we get into the nuts and bolts of how this instrument actually works, we really need to understand its vast, real-world applications. The compound microscope is pretty much the unsung hero of modern society. In medical diagnosis, pathologists rely on it every single day to identify cellular abnormalities and diagnose diseases. Over in material science, it lets researchers spot microscopic defects in building materials. It is the absolute backbone of forensics for analyzing trace evidence like hair and fibers, and it's essential for environmental sampling, allowing us to screen our drinking water for microplastics or harmful microorganisms. And of course, it's a staple in education, helping budding scientists build their foundational skills. To truly master this instrument, we're going to build our understanding piece by piece. We'll start with the structural foundation, move to illuminating the specimen, then the power of magnification, mastering the focus, and finally preparing a wet mount. Section 1. The structural foundation, or the microscope's backbone. I love thinking about this using what I call the power of threes. Instead of trying to memorize some huge random list of terms, you can group the parts strictly by their core functions. You have your illuminating parts, your magnifying parts, and your mechanical parts. Breaking it down like this creates an instant, easily digestible mental map. So this mechanical backbone is made up of the head, the base, and the arm. The head houses the delicate optical components at the top, while the base provides all that essential stability and houses the light source down at the bottom. And the arm? Well, it just connects the two. Actually, here is a really vital practical tip for you. Whenever you're carrying a microscope across the room, you absolutely must lift it using both hands. You want one hand firmly grasping the arm and the other supporting the base at the exact same time. This maintains its stability and prevents any catastrophic damage to that perfect optical alignment. Moving right along to Section 2, illuminating the specimen and controlling the light. You could have the absolute most expensive lenses in the world, but without the right light, they're completely useless. The iris diaphragm, which sits just under the stage, is the solution to this. It functions very much like the pupil of your own eye. By opening or closing it, you control the exact amount of light reaching your specimen. Now, what's really interesting about this is that if your sample is highly transparent, closing the diaphragm reduces the brightness, sure, but it significantly boosts the contrast, making those faint, hard-to-see details suddenly pop right out at you. Tinkering with that diaphragm's aperture size also lets you optimize something called the depth of field. Basically, this is just the range of distances within your specimen that looks acceptably sharp at any given moment. Because you're often looking at three-dimensional structures or layered samples, narrowing that depth of field lets you focus intensely on one specific ultra-thin layer of your specimen, while blurring out all the distracting layers above or below it. Which brings us to Section 3, the power of magnification. This is where the optical magic happens. So you basically have two primary lenses working together in perfect harmony here. First up is the objective lens, sitting right there above your slide. This is your primary magnifying element. It captures the light coming directly from the specimen and forms a highly detailed, magnified intermediate image. These objectives usually range from 4x power all the way up to 100x power. But the image doesn't stop there. It travels up to the ocular lens, also known as the eyepiece. The ocular lens takes that intermediate image and magnifies it even further, typically by another 10 times, delivering the final, massively enlarged view straight to your eye. Think about that for a second. That means a 100x objective paired with a 10x ocular lens is magnifying your tiny specimen a staggering 1,000 times. Taking a closer look at that ocular lens, if you're using a binocular microscope, the kind with two eyepieces, it usually has a diopter adjustment ring. This is a genuinely brilliant feature because human eyes are rarely perfectly matched. The diopter adjustment compensates for the vision differences between your left and your right eye, ensuring that you don't end up with a massive headache after just 10 minutes of observation. Let's move to Section 4 and see how this builds together. mastering the focus to find that perfect clarity. Okay, so you've got the light, you've got the magnification, but now we actually need the mechanical process of taking that blurry shape and bringing it into perfect, crisp clarity. You do this using the adjustment knobs, which are generally found on the side of the arm. Depending on your exact model, they might sit side by side, or the smaller, fine adjustment knob might be stacked directly on top of the larger, coarse adjustment knob. Now, the absolutely crucial takeaway here is understanding exactly when to use which knob. The coarse adjustment knob moves the stage in rapid, large increments. It is fantastic for initially locating your specimen and getting a rough focus, but you have got to use it with extreme caution. If you use the coarse focus while you're on a high-powered objective, you run a very, very real risk of driving that lens straight through your glass slide, completely crushing your specimen and destroying a wildly expensive lens. Once you switch over to high magnification, you must rely strictly on the fine adjustment knob. It makes microscopic, super precise increments that safely bring those final fine details into optimal clarity. Finally, let's wrap up with Section 5, preparing a wet mount. Time for some practical application. Because let's be real, not every slide is just handed to you pre-made from a factory, right? Lots of times you're going to have to prep a fresh specimen right there in the lab. So let's walk through how you'd make a wet mount of your very own cheek cells. You just start by gently scraping the inside of your cheek with a clean toothpick and then smearing that right onto the middle of a clean glass slide. Next, you add just one single drop of methylene blue stain to the center. This step is absolutely vital because human cells are pretty much colorless. Without the stain, you'd just be staring at transparent blobs against a bright light. The stain chemically binds to the cells, giving them a rich, distinct color. Then you gently lower a cover slip over the droplet and use a little piece of paper towel to draw out any excess liquid so the slide sits perfectly flat on the stage. And boom, just like that, you are ready to explore. It really is kind of profound when you step back and think about it. Armed with a solid understanding of its structure, focused light, compounding lenses, and just a single drop of stain, you hold the keys to a completely different dimension. So the only question left to ask is, what unseen universe will you discover next?